Okay, so today we are, oh, seriously, don't be like that. That's not very nice. The chat disappeared. Hopefully, it will come back. So today we are going to start off by talking about the structure of DNA. So it's going to be a little bit of actually stuff that we will pull out from chapter 2, as well as we talk about the basic units that make up the DNA, how it is put together. Then we are going to talk about the structure of our eukaryotic chromosomes inside the nucleus and how those chromosomes are packed in that tiny space inside the nucleus through regulation of chromosome structure. So a lot of, all of us at this point know about how we identified the genetic material as DNA, but that came through a lot of a string of research. Initially, we just had this observation from Mendel, who postulated that there must be some type of genes, but he didn't know exactly what their composition was that were controlling all these phenotypes that we saw in living things. Then we had other scientists like Mesher that studied uh, cells or particles that were coming in pus from surgical bandages and considered that those fragments of cells and those nucleon, as he called them, were DNA. But then later on, as he studied them further, he realized that they couldn't have worked as a hereditary material. So he changed his mind. But then between 1910 and 1940s, a lot of research was done to examine both proteins as well as the nucleic acids, DNA and RNA as genetic materials. A lot of people at that time thought that proteins were more likely to be that hereditary material because there were 20 amino acids compared to just 4 nucleotides, which made them think that, well, we, as complex as we are as organisms, should have a lot of letters in our information book. So then we had the set of experiments that actually helped us figure out what part it was that helped pass on this information. So to begin with, we had the Griffith experiments, where he took a strain from a bacteria that the mice were very that killed mice. So this was the S strain of the S pneumoniae. He injected it directly into the mice, and the mice got infection and died. He then took a second strain, which was known to not cause disease in mice, the R strain, and he injected that into the mice, and the mouse continued to live and didn't have any issues. He then took the S strain and then heat treated it to kill any particles that were any living cells that were there with the help of heat. When he injected those, that mixture then into the mouse continued to live since there were no living bacteria in there. Finally, he mixed living R strain inside the tube, which had already been killed, where all the bacteria had been killed using the heat, and injected that mixture into the mice. And not only did the mice die, but also when he looked at their bloodstream, he found that there were living actual S strain inside those mice. That meant that whatever was in that mixture was actually strong enough to avoid heat, one, right, to get destroyed by heat, and two, that it could then get somehow transferred into those living R strains and cause mice to get infected and die. Now that's very important, right? We all know that proteins lose their structure as they are heated because their bondings are not there, and so they no longer can maintain their functional form. So doing this experiment gives you a clue already that protein is highly unlikely to be the reason for the hereditary information transfer, as we thought. Now, next, we had, multiple groups that then started to focus more on this to verify that yes, it has to be something other than protein that is that is acting as the genetic material. So Avery, McLeod, and McCarty were the ones that were doing these set of experiments. They continued working with the S and the R strain of bacteria, the cells, and they prepared extracts from them so that they could specifically isolate just RNA, just protein, just DNA, just lipids, and just carbohydrates. So they had multiple extracts from these cells to have, isolated fractions or enriched fractions. They wouldn't be perfectly isolated, but enriched fractions from each one of these categories. They then would mix them with the R strain and see if they had the ability to transform the R strain into S strain as they expected. And lo and behold, the only thing that was able to do that was the DNA fraction. Everything else, the cells remained the same. They did not cause any infection, and that was it. So their experiments definitively told that the heritable transforming principle component was in fact the DNA and not anything else.
And that was because the DNA was the only one that was able to mix with it.
Well, so what they did was once they had these fractions, they were mixing them with the R strain to see if they could transform the R strain into the S strain. And the only thing that transformed it into the virulent kind was the DNA. Lipids added to R strain had no effect on them. They still remained non-infectious. Carbohydrates, it still didn't cause any infection. Protein still didn't do it. RNA didn't. Only DNA, when added to the R strain, could transform them into the S strain, where they could then be virulent and cause infection. Does that make sense?
Yeah, thank you.
So in the Griffith experiment, the heat was not enough to kill the DNA?
So yeah, so think about this, right? Think about Jurassic Park, for example, or all the studies that we do where we look evolutionarily in the DNA sequences that come out. DNA is actually a quite stable molecule. Protein can get just, RNA, you sneeze on it funny and it destroys, right? RNA is a very unstable. Protein are, because their whole function is dependent on those hydrogen bonds and the, interaction between different R chains to make that 3D structure, is again relatively sensitive to a lot of environmental factors, including heat, which causes those bonds to separate and make it into its long chain, get denatured instead of stay compact in that 3D stage. So DNA, on the other hand, we can even find millions of year-old fragments of DNA in fossilized structures, right? So DNA is relatively stable and is not going to be destroyed by simple heat. An example, one way to remember maybe with this anecdote is that I actually had this gene that I was cloning, and I cloned it into a plasmid, which I was going to use to put inside the cells of interest. I lost my tube and I looked everywhere. I could not find it. And I remade it. I did my work. And then one day I was cleaning around my area. This is like 6 months later. And I found a tube underneath my bench. And that was it. I tested it. It still worked. No issues. So DNA is not going to be destroyed very easily, as in typical environments. So just a little bit of heat wouldn't be enough to destroy it, while it would still be enough to destroy the proteins and RNA components that are inside the cells. Okay?
So in the third experiment of the mouse, when they only heat the S strain,
Yes.
why then the mouse didn't die? Because like the proteins.
So here they just heated the S strain, right? So they kill the cells essentially. The cells are dead, but the DNA is fine. So in that mixture that they got at the end, the DNA would still be intact, but the membranes of the cells have destroyed, the proteins have denatured. So when they injected, that DNA just by itself can't do anything unless it was inside an actual living cell. Does that make sense? Like it can't go in the mouse's cells and cause it to turn into a infectious bacteria, because those cells can't do that. But it can go inside a living R strain cell and turn that one into a virulent kind.
Okay, got it.
Okay, so because the cells died when you heated them, the DNA was intact, but the cells were dead because the lipids membranes had completely disintegrated, the proteins had denatured, so there was no cells to cause infections. Here, after they heated it, they mixed it with living R cells, so there were cells that could turn into the virulent kind and cause infection. Okay. Okay, so then now that they know that yes, DNA does seem to be the reason, you still, in science, you will notice you need a lot of proof, many different ways before it is considered to be actually the case. Because once you have an idea in general, in everybody's head, it isn't easy to change that thinking, right? A lot of people need to get different ways of proving the same idea. So then Hershey and Chase were the ones that worked on this next. And they worked on it differently. So see, up till now, they were doing all this work in the same kind of bacterial strains. So there wasn't, there was one model system they were using. They still needed to use other model systems to show this as well. So this time they used a specific virus that was made entirely, it's a DNA virus, it's made entirely of just protein coat and DNA. That's it. That's the total component. And this virus can infect bacterial particles. So what they did was they took one mixture of viruses, one suspension of viruses, and they had it in the presence of P32 or phosphorus that's labeled with 32, radioactive phosphorus, so that they could label all the DNA that was getting made in there with that phosphorus. The second one, they had it in an environment where there was the sulfur was radioactive. Now, sulfur is not found in DNA, so you're not going to get any of the DNA labeled. Only the protein is going to get labeled in that case. And in the case of your DNA as well, the first case as well, if there's only phosphorus that's radioactive, it's not going to label any of the proteins, the side chains, because they don't contain that. And then they mix that with the bacteria inside a tube and allow them to infect the bacterial particles. Once they were done with the infection, they kind of blended them briefly, just long enough not to destroy everything in there, but just a little pulse in order to open up the viral heads so that all the stuff comes off and they can look at it further. Now they looked, then they isolated, they pelleted the bacteria, and they looked at what they found inside the bacteria, whether they found the protein component of the virus or if they found the DNA component of the virus. Now, remember, once the virus has infected the bacteria, there will be some protein getting made, but it shouldn't be the radiolabeled kind. And what they found is that majority of the stuff they found in the infected bacteria was actually the P32, the radiolabeled phosphorus, which meant the DNA component was the part of virus that infected these bacteria and not the protein inside. So that definitively then told that yes, DNA is the heritable component. That is not only the heritable component, but also the infective component for these in this case. Does that make sense? Questions on this experiment?
How did they label them with the radioactive isotopes?
So they had them in environments initially all by themselves where they either had them or with actually not by themselves, but in the presence of E. coli where they either had them with phosphorus and radioactive phosphorus only. So anytime they were incorporated, it was incorporating radioactive phosphorus inside their DNA. Anytime there was replication of that. And in the second case, they had them in an environment where they had radioactive sulfur as the only option available. So any amino acid that used sulfur would have been labeled with radioactivity as it was made. Does that make sense?
Yes, ma'am. Thank you.
Okay, So now let's go back and remind us ourselves what DNA is made up of and what does it look like. So building blocks of DNA essentially are your nucleotides. A nucleotide is where you have the nitrogenous base, which is right here, linked to a sugar, which is then linked to a phosphate. So that is what you would call a nucleotide, which is the basic component of DNA. Now, there are 4 different nucleotides that are available. They are organized into 2 categories. There are 5 total, but 4 of them happen in DNA. So they are labeled either as pyrimidines or as purines. In pyrimidines, you have a single ring structure in the base, and they are uracil, cytosine, and thymine. In DNA, you will not find any uracil. The only ones you'll find would be the cytosine or thymine. And in purines, you have the 2-ring structure, so they are larger in size because of this larger structure. And the 2 that you will find would be adenine and guanine. They are usually abbreviated as A, G, C, U, and T. So that's why we have that A, T, C, G, as the typical labels that we see inside it. Obviously, the nomenclatures can be a little bit confusing. So that's why you want to remember that the nucleotide, it is only going to be called a nucleotide if it has the base, sugar, and a phosphate all together. If it's only the sugar and the base together, it is what we call a nucleoside. So it isn't called a nucleotide until the phosphate is added to it. The other thing we want to remember is that these bases, in combination with phosphate, can be found in multiple ways. They can have 1, 2, or 3 phosphate groups attached. So when we write them out, you will either have a monophosphate, diphosphate, and or triphosphate to signify how many phosphates are attached to the nucleoside base unit, right? So if it's a single phosphate, it is what you call adenosine monophosphate, or guanosine guanine monophosphate, whatever the case may be. And if there are 2, it would be di. If it's 3, like ATP, it would be triphosphate. Questions on that?
Is the 1, 2, or 3 phosphate group only for the nucleoside, or can that be for the nucleotide as well?
It can be taken any of the ways, but usually the phosphate group, because it needs to attach to the sugar, it's not going to attach to the base by itself. Does that make sense?
Can you repeat that? I'm sorry.
The phosphate is not going to attach to the nucleoside, the nucleoside, the phosphate can't attach to the base by itself. So it needs a nucleoside before you will attach a phosphate. And as soon as there is a phosphate attached, it's a nucleotide at that point.
Okay. Okay.
Does that make sense?
Yeah. Thank you.
Okay. So yeah, so the bases are adenine, guanine, cytosine, and thymine. When they turn into a nucleoside, which means they are attached to a sugar, they'll be called adenosine, guanosine, cytidine, uridine, thymidine. And then once you have the phosphate attached, that's when you add that mono, di, or triphosphate to the abbreviation nomenclature. Okay, so how are they made into DNA now that we have actual nucleotides? Well, if you look at a nucleotide structure, you notice that you have the nucleotide that's going to be attached to the sugar at the first carbon. And then there are 2 other places that you can have attachments to, which is the 3' carbon and the 5' carbon that are open. The phosphate is always going to bind to the 5' carbon from on the sugar, and that phosphate will attach to the neighboring nucleotide on its 3' carbon. So the bond between the 2 nucleotides happen at the 3' to 5' carbon location, okay? And it's a specific bond. It's called the phosphodiester bond, where the phosphate is binding on the 2 sides, with one side binding to the 3' carbon, the second binding to the 5' carbon. And that is the way it will work, both in DNA and in RNA. This gives your DNA strand a polarity in a way, right? So you have a 5' open end with the phosphate and the 3' open end where you have your the 3' carbon open for interaction. That's similar to, any molecule, any of these polymers that you have looked at. You saw the same thing happening in protein, where you have the amino terminal and the carboxyl terminal. Here, it's the 5' end versus the 3' end. Yes, it is. I just saw the chat message. Oh. So now let's look at what other function these nucleotides can have. So we all know that it can make, obviously, the DNA and the RNA, and we'll look at that further. But remember, because it can attach these phosphates in chain rather than just a single phosphate, as it does in DNA and RNA, it can also act as a short-term energy unit for the cell. And actually, that is our energy currency, right? At the end of the day, that's what our cells use to produce energy or use energy all the time. So in the adenine, adenosine triphosphate, where there are 3 phosphates attached to it, that phosphate group can be pulled off for energy, and then the remaining ADP can then go in and get another phosphate and repeat this process again. So this triphosphate, anytime you have a triphosphate nucleotide, that is energy that's available for cell to work during its metabolic pathway as it works through those. Nucleotides also, so nucleotides have obviously many functions. One of them is as information storage. That's its main job in our nucleus, for example, in the form of DNA. That's also the case in the form of RNA. It's essentially an information storage molecule. It can be used for energy consumption or energy carrier in the form of ATP and GTP, which is where they have the triphosphate that can be broken down. These individual bonds can be broken down to get release energy through hydrolysis of this molecule. It can also combine with other enzymes and coenzymes to form functional groups, actual functional groups within the protein molecules. That's an example of that is coenzyme A, where it is only functional once these phosphate groups are there attached to it for its use. And then finally, it can act as signaling molecule. So when these phosphates are removed, you can create the intermediates like this AMP or ADP. Those can be used as signaling molecules between different segments of the pathway, as in the case of cyclic AMP. Okay, so let's go back now and look at DNA's secondary structure. We know what it's basically made of. Let's go back and see how do we combine that together. So again, several different experiments were done over a course of a decade to get to the point where they were able to actually see that DNA secondary structure and how it functions. So chemists knew by the time that we got those Chargaff experiments done, chemists knew that if we looked at the DNA as a structure, they knew that the backbone of DNA was made of sugar and phosphates, and they knew the basic structure of nucleotides. So in this case, you can see how multiple disciplines of science had to come together to get you the full picture. A lot of the work that was initially done on these structures were actually done by chemists and not by biologists. And then another chemist, Chargaff, established that if we broke down any fragment of DNA, he always found that the total number of purines that were present in that fragment were equal to number of pyrimidines that was present in the sequence. And furthermore, he was also able to delineate that if we looked at individual nucleotides that were there, individual units of these in there, he always found equal numbers of T's and A's and equal numbers of C's and G's. So again, that was something, that was already known by the chemists, but then that was not put together as a final structure until we got that Watson and Crick model at the end of the day. Then Rosalind Franklin and Wilkins were using X-ray crystallography to look at DNA structure more in detail to try to delineate its structure. Through their famous photo 51, Franklin's photo 51, they were able to know that it was some type of a helical structure They couldn't tell if it was 2 or 3 strands that were wound together, but they knew that it was composed of a helical structure. And the reason for that is that when you look at that picture, you can see this kind of bending in the shape and stacking on top of each other, which is indicative of the the sugar phosphate backbone in the back. And then they could see these planar molecules inside, which they could tell would have been the nucleotides. So one of the things they were doing to this DNA, they were looking at it with and without hydration to get different angles of the DNA. They were measuring the distance between individual sugar molecules and the bases, and between 2 sugar molecules, to see if they could delineate that structure a little bit better. So that is how chemistry and crystallography studies were helping with that structure formation. So finally, when it was discovered and put together through the work of all these other people, so Frank, if you look at Watson and Crick, they weren't the ones doing any of the experiments. They were essentially just doing model building, using all the information that they were finding already published or soon to be published by these studies. And so they put that data together that they knew that was already known, and they were able to create a structure that kept all that data constant, that kept all that data to be correct, to form that double strand. So if you look at the DNA strand where it is not in a double strand formation, you will notice that you have equal numbers of C's and G's, equal numbers of A and T's. But to do that, the 2 strands of the DNA have to run in an anti-parallel direction. So one of them is laying 5' to 3', while the second one is going in the opposite direction. So the arrow indicates the side that is the 3' end of that DNA fragment. Now, to keep them in this location, the most energy efficient way, or the way to get these molecules, the individual nucleotides closest to each other, to interact in the hydrogen bonds, is to form this helical structure, so that they are bending in just the right conformation to create those bondings, those interactions, to keep the helix together. The second thing you will is the distance between 2 carbohydrate always has to stay consistent, which is the 0.34 nanometer, and that can only be done in this conformation as you go through the helix. Now, when we look at it more in detail to see what type of bonds are keeping those 2 strands together, you will notice that CG can make 3 hydrogen bonds with each other based on their conformation, while A and T can only make 2, right? Because there are only 2 of those hydrogen bonds that can be formed in their correct orientation, while keeping the width of the helix constant. Now, this is how you would normally see it in a picture, in a 2D picture, but inside the cell, these cells, as they bend to form the helix, you will notice that the actual bases are planar. They are going parallel to the backbone. They are not going in up and down form like they show here, but rather they are flat in a different orientation from the backbone itself. And that allows them to get closest to each other to form the optimal hydrogen bonding interactions between the 2 bases. So things to remember, that they are going to be laying parallel to the backbone, and 2, that they are going to have 3 hydrogen bonds between C and G, and 2 between A and T. So let's say on your test, I give you 2 fragments of DNA. One has a lot of AT components, and one the pairs, and the second has a lot more CG pairs. They're both equal in length otherwise. They're 20 nucleotides. One has 15 of those are CG pairs, and in the second one, 15 of those are AT pairs. If that is the case, which one of these will dissociate from each other first, and why? So if I have those two strands in different tubes and I'm going to heat them to dissociate the two strands of DNA away from each other, which one will melt first?
The one with the A and T, because they don't have enough hydrogen bonds.
Exactly. The one with AT is going to melt first because it won't have as many hydrogen bonds. Exactly. Okay. So just again, reviewing that, there are two chains of DNA that are coiled around a common axis. Their width is going to be constant throughout, right? The helix has 2 nanometer in width. Inside, you are going to see that it is twisting, right, in this double helix. And that conformation will give you a repeated pattern of a major groove and a minor groove, where there's a larger loop of the DNA nucleotides followed by a tighter loop. The sugar phosphate backbone is on the outside. In this model, all the black are the carbon, the white, this is the ball and stick model, so your or space filling model, and the whites are your hydrogens, right? And then the nucleotides, which contain the nitrogen, are the nitrogen is shown in blue, are facing inside. So they are towards the inside of the double helix. The bases are stacked on the inside in a planar manner, in a flat manner, while the molecules in the backbone are in a different orientation. So everyone good? And the strands are running anti-parallel. So it's 5' to 3' on one side and 3' to 5' on the second strand. Questions about this one?
Just to make sure, the phosphate is the yellow, right?
Yes, the phosphate is the yellow. Is that the only color I didn't explain? Yes,
Got it.
the phosphate is the yellow. Red are the oxygens, I believe. Yes, they look like oxygens. Okay, so, well, that's what I just did. I should have. So the two strands are held together by hydrogen bonds between the pyrimides and purines. You're always going to have the complementary base pairing was something that Watson and Crick actually showed in their structure. So while Chargaff had figured out that there were equal amounts of these, he hadn't gotten this together, that they would actually pair with each other. And so that is something that Watson and Crick did come up with as they were building the model. They realized that that's what needed to happen to keep that rule intact and to keep the width of the DNA intact all throughout the strand. So A and T will always pair together, C and G will always pair together. The strands are going to run anti-parallel, and the sugar phosphate is on the exterior, nitrogen bases are on the interior. So now once we are inside the nucleus in an everyday life scenario, the DNA is going to look very different. Our eukaryotic DNA is combined into chromosomes, it's packaged into multiple chromosomes. Depending on the organism, that chromosome number is going to be intact, obviously, but it will be different for every organism. So in this particular image, you're shown a technique called chromosome painting, in which, the chromosomes have been painted by using complementary strands of single-stranded DNA that focus on individual chromosomes that are found in that are unique to each individual chromosome that's linked to a fluorescent molecule of a different color, so-called. Now, when you do this technique, you partially open up the DNA in your chromosomes and then hybridize your little linkers to them, because once you open them up, then they can find the complementary base pairs and bind to them in order to get them to fluoresce in a particular way. So then once you do the procedure, you lyse open the cells so that you can get to the DNA to look at it. And so this is right after the cells have been burst open. And when you look at them initially, you're just going to see them laying out like this. That's how you're going to find them. They're not going to line up all perfectly like this. This is created from this image artificially by kind of cutting and pasting them in their right position. So when you
I'm sorry, will that kill the chromosomes when they fluoresce like that?
Well, you kill the cell because you burst open the cell, right? So
No, never mind.
yeah, the cells are dead.
Brain fart. Never mind.
Yes, cells are gone. So now you just line them up so you can look at them and you can look at how many are there, what general shape is there, right? So you can see the general structure of that chromosome, and each chromosome is going to be different size. The kinetochore is going to be at a different place. The orientation will be different. So you will see the various sizes. They'll always be in pairs because you have one from your mom and one from your dad. So in a eukaryotic chromosomes, at least you'll see these pairs of chromosomes. So this eukaryotic DNA is housed in the nucleus in the form of multiple chromosomes. Our genome contains a lot of nucleotides, 3.2 billion nucleotides, and they are packaged into 24 chromosomes. Now these chromosomes, you don't always see them like this inside a cell, right? To get this picture to happen, you actually have to catch the cell in mitosis when these chromosomes are actually visible. Because normally the chromosomes are not present in this form, and instead they are present in the form of chromatin, which is a very different way to package the same DNA. It's a complex of DNA and protein that organizes them into areas that are open and areas that are closed according to the needs of the cell, and are fitted inside that tiny nucleus in the right way. The chromosomes that you have from your two parents are called homologous chromosomes. So in this picture, these two would be called homologous chromosomes, one from each of your parents. These two are homologous chromosomes within each set. Now we can use the technique of chromosome painting not only to look at individual chromosomes, but to actually look for issues with the chromosomes in a particular person that may have some inherited genetic disease, or that may be coming up with a disease like cancer afterwards. So abnormal chromosomes, whether it is trisomy 13, where you have 3 copies of chromosome 13, or another one is going to is sometimes found using this exact technique, where they're looking at abnormal chromosomes, either number or structure, through chromosome painting. In cancer, this is a karyotype. So by the way, that's something I didn't mention before. When you line your chromosomes up after chromosome painting like this, it is called a karyotype. So this would be an example of a normal human karyotype, where you just have 2 copies of each chromosome. They look the same, they are behaving the same. You don't see any major deletions, substitutions, additions, duplications in there. This is an example of a karyotype taken from an aggressive cancer cell. And you can see that you have multiple chromosomes that are no longer working the way they should. You have chromosomes that are cut and pasted from each other. So there are complete translocations. There are places where there are duplications of chromosomes. There are places where there are fragments taken off of them. So these are examples of extreme conditions where it is a super aggressive cancer for sure. Now these chromosomes can, yes, question?
Aren't there chromosomes? Aren't there 23 chromosomes that the nucleotides are packaged in? Not 24.
So there are your yes, there are 23. I'm sorry. I'm completely there. So yes. There are 22 plus your sex chromosomes, so 23 pairs. I apologize. That I knew, but just a mistake. Okay, so then chromosomes are put together. When you look at the chromosomes in detail, you will find that there are large areas of chromosomes that don't contain any actual gene information, while there are other areas that contain gene information or actual recipes for genes. Now, this extra area is not just junk DNA. Some of it is not used, but some of it actually contains regulatory elements. They contain areas that are going to affect expression of other genes downstream or upstream of them. So those are important areas as well. Now, this image is showing a subsection of the yeast genome, a 0.5% section. And you can see that in that region, there are multiple genes that go in either orientation on those 2 strands. So both strands are not coding for the same information. On one strand, there are a lot of genes in this case. The other strand has a fewer ones, but there are different areas of each chromosome, each side of that chromosome, each side of that DNA that code for genes. So these individual segments that are in orange over here are what contains the genes that instruct either for protein production or for an RNA molecule. Majority of the genes in our body or in our genome are going to code for actual protein products. A minority of them code for regulatory RNAs or other RNAs that are going to be incorporated as a part of protein to get functional proteins as well, or that work all by themselves on their own. So majority of the genes, like I said, will code proteins. A minority of them will have RNA. Now, these are found inside the nucleus as long, they are packaged inside the nucleus because there's a lot of this DNA in these nucleosomes, which are the basic units for eukaryotic structure. So if you look at your chromatin and you stretch it out, you will see that it shows up as this thick threads. If you denature them further, you will see this bead on thread formation. This is individual beads that you see are actually histone molecules, which is an octamer of protein subunits joined together, kind of looks like a yo-yo, that has the DNA wrapped around it. And the strand that you're seeing is the DNA itself. If you look at it closer in this cartoon, this is what you will see. The DNA is wrapped about twice around each one of these nucleosomes that contains the histones. And then there's a linker DNA in between them, and then it continues. If you cut it in between each one of these linker DNAs, you will find that there's approximately 200 nucleotide sequences in this circle plus the linker. However, if we use a nuclease to digest this in-between linker and just take the DNA that's wrapped around the nucleosome, because the nucleosome actually protected from getting digested by these nucleases, you will end up with a fragment of DNA that's 147 nucleotides in length. So each histone octamer essentially wraps about 147 nucleotides of DNA on there. Okay. If we look at the histone itself, you will notice that it is composed of 4 different types of polypeptide chains, right? So this is histone, and each one of them are in dimers present inside this histone complex, in this nucleosomal complex. So you have 2 histone H2A, 2 histone H2B, 2H3, and 2H4. Each one of these have a globular part that interacts with other proteins, other histones around it, and then it has a linker chain that comes out. These N-terminal chains are going to be modified in order to get, give essentially, just like proteins are modified to tell the cell where it needs to go, these tails are modified to tell the genome whether it's going to be open or tightly packaged. So here we're going to look at this a little bit more in detail. So you have your short region of double helix, right? This is a strand, just a double helix. It's going to be wrapped around the nucleosomes to make a long string of chromatin. Once it starts to get wrapped around these nucleosomes, it's called a chromatin. These nucleosomes are then kind of twisted around each other to make a tightly packed structure. That's the chromatin fibers that are then found inside your DNA, inside your nucleus, looping in and out of that space inside the nucleus. Those, when they condense further, are going to create the chromosomes. So to make the chromosomes, they will kind of wrap on top of each other and condense further to create the mitotic chromosome. Okay, so at the end of the day, by the time they get fully condensed, they have been essentially condensed to 10,000 their normal size. So how this happens is through chromatin remodeling complex, which is a set of proteins that work to locally either wrap or unwrap the DNA as the need arises. And that's done by putting tags on those tails of nucleosomes. So the DNA, whether it is twisting them around the nucleosome or unwrapping it, is going to require ATP hydrolysis. And these complexes will essentially cradle the nucleosome, fit it inside its globular, active binding site, so that it can then twist around it to move the DNA, either close it or to open it. Either one of that is going to require ATP hydrolysis to move along this fragment and wrap it or unwrap it. And it takes several steps of this hydrolysis to get a small segment to unwrap or wrap. So how it does that, how is it recruited there, is, like I said, based on the modifications that are present on those tails. Now, the several things are going to be modified inside the final DNA. The DNA itself can get modified as well, and the nucleosomes and histone and tails can modify it as well. In a normal closed conformation of DNA, which is called heterochromatin, which is tightly packed DNA, the histone tails are going to be modified on the outside to wrap around the DNA and keep it tightly in place. Those tails are going to be, those modifications are going to recruit these units to come in, and they will continue these modification process. The heterochromatin-specific proteins that bind to it will continue to create more modifications of that kind on the adjacent DNA until they reach a barrier sequence where there is essentially like a linker sequence where there is no modification present. And that essentially is a signal for them to fall off. So the modifications that are present on the heterochromatin cause more modifications to happen on adjacent DNA until they reach an open space where gene expression is happening. And that is going to then stop this complex from going further and fall off. Now, here again is an image, a cartoon of the nucleosome. You see that you have the globular parts all interacting with each other in that little tetramer form. It's an octet at the end of the day. There are 8 units together to form the nucleosome, and the tails are hanging off. So here you can see this cheese-looking shape is the globular portion of the histone, and then this is a portion of the N-terminal tail. If you look at the individual amino acid residues inside it, you will see that there are multiple residues, specific residues, that can get modified in that space, okay? So this is looking at histone H3, which is one of the most modified histone inside our nucleus or inside the genome. And you will see that there are, arginine and lysines. Lysines especially are highly prone to getting modified. These can get either phosphorylated, methylated, or acetylated. So there are 3 different types of modifications that can happen in this tail region. Methylation, typically you would know, would shut down the DNA expression, the gene expression in that area, whether it is methylation of your protein strand or methylation of the DNA strand itself. But it only happens if that's the only thing that's present. So here is an example where you have heterochromatin. It's going to be heavily methylated many times by 3 methyl groups on each one of these sites. And the more heavily methylated it is, the more tightly it's going to condense and close that area to silence the gene expression in that area. On the other hand, you may instead have acetylation of that same residue when it is required for gene expression. In that case, it doesn't mean that there's no methylation around. There'll still be methylation at other sites on that same N-terminal tail, but the key points that are required for activation of gene expression will be acetylated at that point. So this residue, for example, K9, the 9th lysine in this sequence, can be methylated or acetylated. It's never going to be both at the same time. If it's methylated, it is silencing gene expression. If it's acetylated, it's activating gene expression. The more residues that are acetylated, the more activation of the gene underneath it is going to happen. The more loosely it's binding to that sequence, and the more open the genome is at that spot. Is that clear?
So when the genome is more open, it means that it can express freely?
If it is open, it is more open for transcription factors to bind to it and express the genes that are present in that space. Now, that's what I meant, that maybe only one of these is acetylated. So it's open, but it's not fully open. And so transcription factors will bind to it, but at a very low rate. But if there are 3 of these acetylations back to back to back, it's going to be more open in that space and more transcription can happen. So it's a way to regulate not only whether the gene is expressing, but also how much it is expressing. Whether it's expressing, 1 mRNA molecule per minute or 100 mRNA molecule per minute. Depends on how open or closed that space is. Does that make sense?
Yes, it does.
So that's actually all for today. This one was a shorter chapter, but we had some time, so I can answer questions.
What was the third one? There was methylation, acetylation, and what?
And phosphorylation.
Thank you.
You're welcome. Should I keep the share screen open so you can ask questions?
So like, is it something like if someone got a brown eyes, is it because one of the genes are more methylated from the father's side, maybe? Like, is it that kind of
Well,
stuff?
not exactly. So if a common thing with chromatin is, right, the one that you would all know about is the bar body, which is in the females with 2 X chromosomes. One of the X chromosome is randomly shut down in each one of our cells to keep the important gene expression, the stuff that shouldn't be doubled up essentially, inside our each one of our cells intact. So you always want to have the same amount of gene expression from that X chromosome, whether it is a male or a female, for those essential genes. Well, those bar bodies that form, they form randomly. So let's say that it did contain a gene for eyes color, then it's randomly shutting down either the blue or the brown eyes. It's not that it's only shutting down one of them, right? So that's one example. But usually the eye color is a more complex gene expression. You are expressing both pigments or a pigment and no pigment all the time, but it's which one is the stronger pigment that will show up at the end of the day. So that's not usually associated with methylation of that one gene. This
Okay,
is
got
more
it.
to regulate actual areas of genome that need to be just shut down unless something happens that causes it to be activated and express the genes in that area.
Professor, could you go over again how acetylase and methylation and phosphorylation are regulated?
From this one?
Yeah.
Oh, not this one, the next one, sorry, this one. So what regulates these is actually something we will talk about next week. We didn't quite talk about that. Is other environmental cues that will regulate. One of the ways methylation is regulated is actually just passed on from one generation to the next. So the methylation is something that is maintained through generations, and it is going to be maintained not just on the DNA, but in these areas as well. But there are other types of these specific methylation and acetylations and phosphorylations that require environmental cues. So only when a growth factor binds to its receptor will it open up that particular region by causing acetylation or methylation at these specific residues to open gene expression in that area. And it's not going to be all over. It's only going to be in a very specific space. Okay.
Got it.
So
Thank you.
imprinting is one of the examples that's, and we'll actually talk about it maybe next week or next class. When we do chromatin remodeling, we'll talk about imprinting, which is how methylation is transferred one generation to the next. And that one is the only one which is maintained exactly the way it was before. And depending on the species, sometimes it's coming just from the males and sometimes it's coming just from the females. Okay.
Thank you.
So when you say that it's open the gene expression, this is the histone tails, like it's open the DNA strands?
Yes, so look over here. So the nucleosome, right? So you have the yo-yo and the DNA is wrapped around the yo-yo. And then you have, how you have the string of the yo-yo? So it would be as if that tail is wrapping further on top of that DNA strand to keep it in place. So by methylating it, that binding is more tight. By acetylating it, it's opening up. So that particular area is not binding to the DNA as well. And now that DNA can be a little bit more loose in that space.
Is it that mean all the important genes are wrapped around these histones?
Not all so some areas, some housekeeping genes are always active. So they would be most likely in these barrier sequences or in the, in open spaces where there aren't nucleosomes always bound. Even when you look inside the nucleus, you will see, and I may have a picture of that. Maybe not. If not, I will bring one. You will see areas inside the nucleus that are more tightly looped and others that are more loosely looped. The areas that are more loosely looped are reflective of areas that are doing gene expression in there at that time, that are open. They won't have nucleosomes around them. They won't have anything else, and they're opening up to do gene expression. Okay?
Okay.
So
Specifically,
at any given time, there are thousands of genes in your actual genome. At any given time, not all of them are going to be active. So the subset that's active is not wrapped. The rest of them are the ones that are wrapped around nucleosomes. In addition, there are two different types of this chromatin. You have heterochromatin, which is more tightly packed, and that's indicative of areas that are either not used at all or areas that are very rarely used. And then there are areas of the genome that are going to be more loosely wrapped, the typical wrap around the nucleosomes. And those are areas that are going to be activated more often. Now, the retinal cells or cells in our eyes are going to have different areas of heterochromatin versus euchromatin. This area that's active is called euchromatin, that is replicating and dividing, or not replicating, that is transcribing and translating into proteins. And that euchromatin is going to be different for a hepatocyte versus a retinal cell versus a neuron, because different genes need to be expressed in each one of those places to do the function they need to do. Okay?
Got it. Thank you.
so for this slide, so the barrier DNA sequence is just a way to it's essentially telling the body to stop transcribing or stop transcription or anything like that.
Well, usually those areas will contain actively transcribing genes. So what the barrier sequence in this case is doing is telling this complex, the histone modification complex, the heterochromatin-specific proteins, to stop making more heterochromatin, to stop compacting the DNA. Because here, there is no nucleosome that it can bind to, that it can jump to continue that modification. You see how you have this entire region of euchromatin, and you have in there this barrier DNA sequence. These are modified that bind, that is what the heterochromatin specific proteins bind to. If it finds that this space is open next to it, there's no transcription factor bound, there are nucleosomes there, but it's not, it's not opened up, there is no replication happening, no transcription happening, it will continue to modify that and wrap it up and condense it further and tightly pack it until it reaches a space which is open and has no nucleosome available. that will then signal it in the sense that there's nothing for it to bind to modify. So see how it continued to do it and wrap things up and tightly compact it until it got to the point where it couldn't do it anymore. There's nothing to bind there. So it will fall off the DNA at that point, and the rest of it will remain open.
Got it. Thank you.
So when you're adding all these modifications to the DNA sequences, are you adding them to the genes or the promoter regions or both?
Both type of modifications happen. Right now, we are only looking at heterochromatin made through specific modifications of histones. So right now, we are only looking at these N-terminal tails of the histones. Next lecture, we will look at more the actual DNA modifications.
All right, thank you.
So yeah, so the histone, these heterochromatin specific proteins, they are modifying the histones themselves. Those are the modifications they're putting. Any other questions?
I'm sorry. I think I missed this or something, but the histone modifications, what is the purpose of them getting more tightly packed together versus like being expanded?
So when they're more tightly packed, that area is going to have silencing of the gene expression for that area of genome. Those genes are not going to be transcribing at all at that time, or very little at that time. The areas that are more open are areas that are going to be transcribing to make proteins or RNA that will do the functional work for that cell.
Got it.
So euchromatin is where you'll have active gene expression happening, and heterochromatin is where gene is silenced. Gene expression is silenced in majority of the cases. Always there's a caveat, but that's in general.
It's like the stuff that we don't need in life, we like package them in boxes and put them away.
That's right. So it's essentially the same idea that whatever you don't need right now, you package it tightly and put it away to clean up and create space for the stuff that you do need. Because our nucleus is tiny for all the stuff that needs to be packaged in there. If the whole chromosomes, all the chromosomes were all opened up, all 23 of them were hanging out, in their open state, they can't, there's just not enough space. And two, nothing could get done. So to keeping them packaged is essential for the cell to open up just the areas that it needs to transcribe and translate into actual functional proteins. Okay?
Yes, everything is clear, and thank you so much.
No problem. So yeah, so that's what you saw here, right? So when it's fully opened up, that's when you are transcribing. This is just the first level of packaging that they are wrapping around the nucleosomes. So they're going from, this long strand to a smaller area, and then they're wrapping further around each other to package them tighter. Now, again, those nucleosomal tails, those histone tails, will be the ones helping this packaging along to keep it as tightly packed as possible, depending on how much this area is needed. And then those are then looped in, and those loops are what you find along the nuclear inner membrane. That's where they're organizing themselves. There'll be areas that are going to be opened up there, and those will be areas that are transcribing and areas that are going to be tightly packed. And then during mitosis, each one of those chromosomes will separate and just kind of condense to their entire mitotic full chromosome stage. Okay, guys, so next time you'll probably have your test results back as well. So we'll spend a little bit of time either at the beginning or end going over any big questions or any question that I see like everyone is getting wrong. So I'll go over those with you guys, and then we'll do the rest. We'll do the next chapter next time. Okay.
Thank you.
Welcome.